I'm Eric Brown, professor of biochemistry at McMaster and CEO of Synmedix, a new startup in the area of antibacterial therapies. We've all seen the headlines, such as that shown here in The Economist. There's a desperate need for new antibiotics. Antibiotic drug resistance is one of the biggest threats to human health. It's due in large part to a lack of innovation. Current therapies are based on discoveries that are often made more than 50 years ago, and these are just not suited for complex infections that we're seeing today. This technology was discovered in my lab. We've been working on antibiotic drug discovery for more than 20 years. This is probably the simplest but most powerful invention that we've come up with. We've discovered that bicarbonate, yes, that's the active ingredient in baking soda, enhances and expands the activity of antibiotics. This is the only data slide that I'll show today. For aficionados, they'll recognize that this is a MIC90 determination that shows the activity of azithromycin alone or azithromycin plus bicarbonate against 2 superbugs, 100 strains of MRSA, and 100 strains of Pseudomonas aeruginosa. In all, the data suggests that there's just a massive enhancement of the activity of azithromycin in vitro against these 2 superbugs. We see extended spectrum in that Pseudomonas aeruginosa is not a bug that's supposed to be treated by an antibiotic like azithromycin, and reversal of resistance in both MRSA and Pseudomonas aeruginosa. We also have preclinical validation that I don't have time to show you here, mouse models of skin infection and topical use of bicarbonate and azithromycin to treat them, as well as ex vivo models in human skin. We've done also quite a bit of work on the mechanism here. And it turns out that bicarbonate perturbs the bioenergetics of the bacterial cell membrane, and this causes a large increase in the uptake of antibiotics into bacterial cells. We're staged then for success, we think, in the clinic, where we have a clear safety profile, existing antibiotic potentiated by a benign substance like bicarbonate. This positions us well for an accelerated approval pathway like the 505(b)(2) pathway of the FDA. And we have a platform technology that's widely applicable to a variety of antibiotics as well as antiseptics. We're interested in areas of very high market need and clinical need. And to start, we're focused on diabetic foot ulcer infections. This is a devastating condition, 500 million diabetic patients worldwide, about 100 million foot ulcers, and half of those get infections yearly, where 85% of diabetic amputations are a result of foot ulcers. The 5-year mortality rate of a diabetic amputee is second only to lung cancer, so this is a huge health burden. The marketplace is also large and growing. Systemic antibiotics actually have a very small slice of this market. Bioactives, devices, and dressings are a big part of the market, and none of these therapies are proven effective. None are approved by the regulators for treatment. And in fact, the International Working Group on Diabetic Foot has issued a call for topical antibiotics, which is where we come in. We're poised, and we think, to deliver the first antibiotic approved for diabetic foot ulcer. This is a topical preparation we call BCX of bicarbonate and azithromycin. The idea is potent efficacy at the site of infection, facilitate healing, reduce side effects that are otherwise associated with systemic antibiotics, and reduce treatment cost. We anticipate, of course, premium pricing, being first in the class here, and as well, market exclusivity could be guaranteed, of course, by the regulators, along with our intellectual property. The IP is very strong here. We have 2 patent families underway, one in which we have been granted a patent by the USPTO for composition of bicarbonate and azithromycin, and we have another patent family underway, more widely directed at other bioactives. Synmedix, of course, is an R&D company. We understand that the ultimate destination of this technology is with a pharma partner or a specialty pharma group that know the marketplace and can take this to the customer. They're interested in seeing from us these activities: preclinical and IND enabling studies, along the lines of formulation and manufacturing to get us an IND, as well as a phase 2A proof of principle trial that would show success of this therapy in the clinic. Critical milestones, an IND and a phase 2A complete, we'd get a considerable bump in valuation and be positioned for a license or partnership with one of those pharma. I'll add that the prize money from this competition could be very useful in that formulation element. Strong team here with field leading leadership in formulation, in wound healing, chemistry, clinical development, business development. But I want to highlight this fellow here, Dr. Perry Meyer, our medical advisor, leads the Meyer Institute in Hamilton. It's one of Canada's biggest diabetes clinics, 16,000 patient visits per year, and he is super enthusiastic about what this technology could do for his patients. I'll end with the following. We're on the verge of a post-antibiotic era. We're in a world without antibiotics. More than 10 million people would die annually from infection. That's more than currently die from all cancers. We have a technology which is suited to tackling this in a variety of ways, and I've tried to give you a small slice today. Our mission clearly is to prevent this from happening. Thank you, and I look forward to your questions.
Thank you very much, Eric. That was a great job, and I'm sure we have lots of questions. So maybe this time we can start off with Alex. If you could take yourself off mute and kick off the questioning.
Yeah, very impressive presentation. Very well done. And oh, there we are. And thank you for that. So one of the questions I've got for you is, in terms of the discussions you've got with potential pharma partners, have you had any approaches yet to any potential partners, or are you still looking to develop additional information and validation before you make those approaches?
We're definitely interested in those discussions, and we had some really good ones, in particular with one specialty pharma group in Europe who are very interested in the technology.
Mm-hmm. Okay. Right. Did you have another question, Alex?
No, that's fine for now. Thank you.
Okay, John, if you could come off mute there. Great, thanks.
Yeah, thanks, Dr. Brown. Very, very interesting. So I wonder if you could just drill down a little bit on the roadmap as it relates to trials. So I'm correct in assuming that there hasn't been any human clinical trials yet, and so with that, what kind of results? Maybe it's hard to quantify, but what kind of results do you think you'd be looking at when it comes to human clinical trials? Are we talking, again, a previous question I had was, we have this scale of mild to severe diabetic foot ulcers. What would this be indicated for, or is it more focused on the infection?
Yeah, so certainly you would want, it's a really interesting space where, as I mentioned, there are no approved products. So in terms of the clinical trial, we're in the remarkable position of being able to do, say, a placebo-controlled trial, where in that instance, of course, you would focus on kind of the mild case, right? Maybe to moderate, a case where a foot ulcer infection may not necessarily prompt the use of a systemic antibiotic. Of course, the issue with systemic antibiotics is they don't tend to get well to the site of infection because there's poor perfusion of the wound there. The endpoints in a trial like that would surely be things like, colony forming units per mil at the site of infection, things like, whether or not we can see accelerated wound healing. But it's a relatively straightforward trial in terms of, trying to get to those conclusions and endpoints.
Yeah, so I had some questions. I admire you taking on the diabetic foot ulcer space because it's a very, very difficult space. It's difficult to get funded. And I guess that's because it's difficult to find something that works. Is there something you can do to mitigate that risk a little bit early on, to be able to attract investors into this very difficult space?
Yeah, I think that's a great point, Jennifer. There's been some interesting attempts in this area to get this indication. One of them was a company founded in the 90s called McGainen, and it went through more than one name, in fact. The idea is that frogs don't get acne, and that peptides that are on the skin of frogs can prevent infection. And so that company was, developed around frog peptides, essentially as topicals used in to prevent infection and to cure infection. If you look at the fine print of those discovery papers, those peptides were really not much worse at killing human cells than they were at killing bacterial cells. And in fact, they didn't get through in their phase 2 trial. In fact, the placebo arm did better than the treatment arm, which is an indication, I think, of the tox of those compounds. Mm-hmm. So there's an interesting path set out there, and there was a lot of enthusiasm from the diabetic foot community at the time for that. But I take your point that there's some considerable risk. It's a difficult patient population, et cetera. Mm-hmm. We're also really interested in burn as a possible area. It's a much smaller market, of course, but there's also great enthusiasm, from people in those quarters who are trying to, treat some of the most difficult patients. We could also think a lot about things like investigator-initiated trials around people who have things like indwelling devices. Mm-hmm. Spinal surgeries in particular, those wounds are often very difficult to close, and complications with infection are common. We've had inquiries, for example, from people in peds in that realm. So there are, a number of possibilities here, I think, for us. I'm particularly excited about the burn idea, secondarily to the diabetic idea, because those wounds are so similar. Poorly perfused, of course, by the blood, a shortage of bicarbonate at that site, which is otherwise there in human physiology as an extracellular buffer. So it's I think there's more than one possibility here. Mm-hmm. And diabetic foot ulcers may not be the most sexy, but there's a huge medical need, a huge market, and an opportunity to be first in class.
Okay, great. Well, thank you very much, Eric and team. Great presentation, and appreciate your answers to our questions.
Thanks so much for your time. We really appreciate it.